Bismillahirrahmanirrahim. Today the topic of my discussion is pyruvate dehydrogenase complex. It is a multi-enzyme complex that catalyzes the conversion of pyruvate to acetyl-CoA. It is an irreversible step, and we know that the pyruvate is generated from glycolysis. The two molecules of pyruvates are formed, and then pyruvate is converted into the acetyl-CoA. And from acetyl-CoA, the next cycle begins, that is known as TCA cycle, or also known as Krebs cycle. So it is a link between the glycolysis and TCA cycle. How? The glycolysis produces the pyruvate molecule, and then acetyl-CoA is produced, and this acetyl-CoA enters the Krebs cycle. So for the conversion of pyruvate to acetyl-CoA, there is a complex enzyme system that consists of enzyme and coenzyme, that is overall known as pyruvate dehydrogenase complex is used, which converts the pyruvate into the acetyl-CoA. So as acetyl-CoA begins the next cycle, and pyruvate is the molecule of the previous cycle, so from it is a bridge between the two cycles. So for the conversion of the pyruvate to acetyl-CoA, for the formation of the next steps, or for entering into the next steps, and continuing the next cycle, there is a conversion of pyruvate into the acetyl-CoA. This enzymatic complex is used. It is composed of 3 enzymes and 5 coenzymes. 3 enzymes that are apoenzymes. Enzyme 1 is known as pyruvate dehydrogenase itself. Enzyme 2 is known as dihydrolipoyl transacetylase. Enzyme 3 is known as dihydrolipoyl dehydrogenase. The 5 coenzymes are thymine pyrophosphate TPP, derived from vitamin B1, that is thymine. You can also watch my lecture of vitamin B complexes. So in that lecture you will have a detailed description of vitamins B, that what are vitamins B, what are their types, and what are their common names. Actually, thymine is the name of vitamin B1, and when this thymine is in activated form, it is known as thymine pyrophosphate TPP, and it is a coenzyme basically. Second coenzyme is lipoic acid, third is coenzyme A, fourth is FAD, and FAD is also derived from riboflavin, and niacin is also a vitamin B, riboflavin is also vitamin B, and that catalyzes the reaction related to the FAD, and niacin catalyzes the reactions related to the NAD positive. So these are the 5 important coenzyme: FAD, NAD positive, coenzyme A, lipoic acid, and TPP. So these 5 coenzymes form the pyruvate dehydrogenase complex along with these 3 enzymes. So there are total 8 things, 5 coenzymes and 3 enzymes. First of all, we can see that there is a removal of carbon dioxide, that is known as decarboxylation, that is the release of carbon dioxide from pyruvate. So we know that the pyruvate is a 3 carbon compound and acetyl-CoA is a 2 carbon compound. So when 1 carbon dioxide is released from pyruvate, so it is converted into the 2 carbon compound. So carbon dioxide is released, so pyruvate is converted into the 2 carbon compound, that is known as acetyl-CoA. Then hydroxyethyl group is transferred to TPP. First of all, it is a oxidative decarboxylation reaction in which the first is the pyruvate is decarboxylated, releases carbon dioxide by pyruvate dehydrogenase. Hydroxyethyl group is transferred to TPP. Next, hydroxyethyl group is transferred to the coenzyme TPP, which becomes hydroxyethyl TPP, and from hydroxyethyl TPP, this group hydroxyethyl is transferred to lipoic acid, which is converted to acetyl lipomide with the help of second enzyme, that is known as dihydrolipoyl transacetylase. So you can see that So you can see that it is an oxidative carboxylation reaction in which the pyruvate is decarboxylated and it is converted into the 2 carbon compound. Hydroxyethyl group is transferred to the TPP, which is converted to the hydroxyl ethyl TPP. From hydroxyl ethyl TPP, hydroxyl ethyl is transferred to another coenzyme. That means first of all the enzyme pyruvate dehydrogenase is used, then coenzyme TPP used, then coenzyme lipoic acid is used, and then the enzyme dihydrolipoyl transacylase is used. The acetyl group is transferred to coenzyme A sulfhydryl to form acetyl CoA, and there is a formation of dihydrolipoic acid or reduced lipoic acid. So that was all about the pyruvate dehydrogenase complex. If you have any query related to this topic, you can ask me in the comment section. Thank you very much.